Hey guys, Rouge here, and today we're talking about DNA replication, or how do we get more strands of DNA in our cells. If we think back to the cell cycle, we briefly talked about DNA replication during the S phase of interphase, which is right here. Now after S phase, after DNA is replicated, we go to G2, where the cell grows, and then finally we go to this section of the cell cycle, and these two parts, mitosis and cytokinesis, could be summarized as cell division. So why does DNA replication occur before cell division? Well, DNA replication has to occur before because if a cell split without making an extra copy of DNA, then each daughter cell would actually have half the amount of DNA that it needs. So to make sure that each daughter cell is completely identical to the parent, we have to have DNA replication. So here's some key highlights. I'm going to go over the key highlights first, and then I'll show you what DNA replication actually looks like. So first, like I had mentioned, replication ensures each new cell receives a full copy of DNA. Okay. The first step is we're going to have this enzyme. It's called helicase. And remember, anything that ends in this suffix, anything that ends in ase is an enzyme. Okay. So DNA helicase is going to unzip or unwind the strand of DNA. Each strand serves as a template for its complementary side. So basically what that's saying is if one strand has a T, this polymerase is going to add new nucleotides or new complementary bases. So polymerase is going to read it and it's going to say, I have a thymine, I need to add an adenine. Or I have a guanine, I need to add a cytosine, okay? So each strand that was the world's longest bell ever. So each strand serves as a template for DNA replication. And also DNA replication is called semi-conservative. So if you break down this word, semi-conservative, semi means partially, and conservative means to keep. So what you'll see is after DNA replication, each strand is half old and half new. It's not 100% brand new, okay? So at this point right here, I'd like you to go ahead and put this on pause and then add these notes to your notebooks. And here we go. Semi-conservative, each strand is half old, half new. So here's what DNA replication looks like. We have our strand of DNA, and helicase is going to slide down the strand and separate it. So we have our helicase, the enzyme that unzips the DNA strand, and this area right here, this is called the replication fork, and this is where replication is actually going to occur. And actually, there's some images on the side whiteboard. Go ahead and add those to your notebooks as well. Next, we have what's called the DNA polymerase. And the DNA polymerase is reading each, go and change this color real quick. DNA polymerase is reading each strand, so it reads an adenine and it knows to have a thymine. Over here it reads a cytosine and attaches a guanine. So this is what that means when I say that each strand serves as a template. Okay, so that right there reads a thymine. DNA polymerase adds an adenine. Okay, same thing on this side. You have the cytosine. Polymerase adds a guanine. Okay, so DNA polymerase is going to go down both strands, and remember, it's occurring in opposite directions because remember we talked about DNA is anti-parallel. The structure is actually flipped on both sides. If you wanted to review that, you could go back to the DNA structure video. So polymerase is going to continue down. It's going to go down both strands. And if you notice, this side right here, as soon as this thing's done, if you notice this is an old strand, this is an old strand, and this is a new strand. So this is what semi-conservative means. Semi-conservative. Half of each strand is old, half of each strand is new. But now what happens if we get a boo-boo? What happens if we get some damage? So let's take a look at this. Let's say DNA polymerase is going down and accidentally adds an adenine. Or that would actually be a thymine. Let's say polymerase adds a thymine when it should be a guanine. Well, polymerase can actually fix that. It can detect that error, come back, and swap that out for the correct nucleotide. Okay. Now that's under perfect conditions. So let's look at some facts about polymerase. Number one, polymerase adds about 50 nucleotides per second. That's quite fast. Polymerase will make a mistake about once per billion base pairs. So basically, if I were to take your textbook, your biology textbook, and type that out word for word, I would type that out 1,000 times, that whole textbook, before I make one mistake. Okay, so polymerase is pretty accurate. So what are some things that will increase this DNA damage or this mistake in DNA? Too much sunlight, being out in the sun for way too long could increase DNA damage. Smoking, having alcohol, poor health choices, and also not getting enough sleep can increase the chances for getting one of these mistakes. Okay? But it's okay, because we have polymerase that could fix those errors, right? Well, polymerase has to have magnesium. In order to work, polymerase has to have magnesium. Also, too much of those mistakes, too much of those boo-boos can lead to cancer. Okay? So you don't want those mistakes. You want to try to prevent those. So where do we get magnesium from? Magnesium is found in chlorophyll. And chlorophyll is in green plants. So this is one of the reasons why people always say, eat your fruits and vegetables. I know they say that to lots of young children, but there's an actual reason behind this. Chlorophyll has the magnesium your body needs to make polymerase. And again, without that polymerase, you won't be able to fix DNA damage, and that could eventually lead to cancers or mutations. And we will talk about this more next time I see you. Have a good day.